Hello, guys, welcome back to my YouTube channel Excellence Hub. Today I'm going to discuss the topic BAC and YAC library.
Bacterial artificial chromosome, YAG yeast artificial chromosome. This is the general overview how the library is being constructed. These are the cells that contain the DNA as their genetic material that have our gene of interest that we want to be in our recombinant DNA molecule. Firstly, these cells are being digested with restriction enzymes that are going to produce small fragments of this DNA genetic material. Now these DNA fragments are being ligated into the cloning vectors. These bacterial cells, which are the host cells, are being transformed with these vectors. So now these are the bacterial cells that contain our recombinant DNA. Now, vectors. What are vectors? Vectors are the cloning vehicles used to produce recombinant DNA molecule. Various vectors are being used. Now we are going to talk about artificial chromosome vectors. Artificial chromosomes are DNA molecules assembled in vitro from defined constituents that guarantee stable maintenance of large DNA fragments that are being inserted into the vector. Artificial chromosomes are having various applications, such as it is used for genome sequencing programs, it is used for functional characterization of entire genomic regions, used for the transduction of large DNA fragments into human and non-human mammalian cells. There are various artificial chromosome vectors that are used for the construction of recombinant DNA molecules, such as BAC, bacterial artificial chromosome, YAG, yeast artificial chromosome, HAG, human artificial chromosome, PAC, P1 derived bacteriophage artificial chromosome. This is the table. This is the table that depicts various vector system, their host cell and their insert capacity. What do you mean by insert capacity? Insert capacity is the capacity of the vector system to take up the foreign gene into it. Plasmid is host cell is E. coli. Insert capacity is 0.1 to 10 kb. Lambda bacteriophage, its host cell is E. coli and insert capacity is 10 to 20 kb. Cosmids and phosmids, its host cell is E. coli and insert capacity is 35 to 45 kb. Then next is P1 derived bacteriophage, its host cell is E. coli and insert capacity is 80 to 100 kb. Then BAC, bacterial artificial chromosome, its host cell is E. coli and insert capacity is 50 to 300 kb. Then PAC, P1 bacteriophage derived artificial chromosome, its host cell is E. coli and insert capacity is 100 to 300 kb. Then YAG, yeast artificial chromosome, its host cell is yeast and insert capacity is 100 to 2000 kb. Then HAG, human artificial chromosome, its host cell is cultured human cells and insert capacity is less than 2000 kb. Then, now I'm going to discuss about bacterial artificial chromosome. These are the DNA constructs which are based on functional fertility plasmids or F plasmids, which are used for transforming and cloning in bacteria, usually E. coli. F plasmids play crucial role because they contain partition genes that promote the even distribution of plasmids after bacterial cell division. BAC are capable of carrying approx 300 kilobase pair of inserted DNA sequence. But one problem it has is it has low copy number. It means when this vector system is being inserted into the host cell, the daughter cells that are going to be produced, it can be only one or two only. Like other vectors, BAC contains various or genes into it, like origin sequences derived from E. coli plasmid called the F factor, MCS, multiple cloning sites that are restriction sites, selectable markers that are antibiotic resistance, and these are relatively easy to manipulate, move into cells by transformation through electroporation technique. Common gene composition Rep-E for plasmid replication and regulation of copy number. Par A and Par B. These are for partitioning F plasmid DNA to daughter cells during division and ensure stable maintenance of the BAC. Selectable marker for antibiotic resistance. Some BACs also have laxet at the cloning site for blue-white selection. Then T7 and SB6 Phage promoters for transcription of inserted genes. This is the central BAC vector containing different sites in it. This is ORI-S from where the replication process will going to begin. Rep-E site, partitioning A and partitioning B site, selectable marker site, chloramphenicol resistance site. Then this is T7 promoter and sp6 promoter, and this is the cloning site containing different restriction sites in it. This is the overview how the construction of BAC is done. This is the BAC vector containing origin of replication, chloramphenicol resistance site, cloning site, F plasmid site. Cloning site is the site where the large foreign DNA fragment is going to be inserted. So firstly, we need to digest it so as to produce the sticky ends so that the large foreign DNA fragment can be easily inserted into it. So how to cleave this cloning site? Using restriction endonuclease. Using restriction endonuclease, this cloning site is being cleaved, producing the sticky ends. Now, large foreign DNA fragment can easily be ligated into this sticky ends. And these are ligated into the cloning site using DNA ligase. Now, this is recombinant BAC that is obtained. Now this recombinant BAC is going to be transformed into the host cell through the process electroporation. Now various recombinant molecules are obtained. Now we are going to test which molecules are being transformed that contain recombinant DNA molecule and which cells are non-transformed that do not contain these recombinant molecule. This can be done by using plating method in which chloramphenicol resistant cells are being selected. The plate contain agar that have chloramphenicol in it. What does this mean? This means the cells that are being recombinant and it has recombinant BAC in it. In those cells, particularly this chloramphenicol resistant site will going to be disrupted. So those cells will not survive on the plate. But the cells that are not being transformed, that are not recombinant, that do not contain recombinant BAC vector are going to survive on it because they have chloramphenicol resistant site. Genomic. This is the BAC library construction process. Genomic DNA obtained from the cell. Through digestion process, this genomic DNA is being fragmented into small fragments. Then these small fragments inserted into the BAC vectors. Now these BAC vectors, recombinant BAC vectors are being transformed into the host cell. And these host cells, some are recombinant ones, some are non-recombinant. Through plating, Through selectable marker genes, we can identify which cells are being recombinant and which cells are non-recombinant. Various advantages of using BAC. It helps to minimize site of integration effect. This is the phenomena which has been identified as endogenous sequences such as gene coding regions and distal regulatory elements to be destructed and to produce potentially undesirable phenotypes in gene cloning technology. Human genome banks consist of full gene structure that play a very important role in gene regulation. This is also one of the advantages of using BAC vector system. Therefore, the human genome banks will ensure full mRNA processing and splicing when genes are transcribed and produce the full complement of protein isoforms once mRNAs are translated. It can be transfected and expressed in mammalian cell lines, even if transfection efficiency and copy numbers are low. Various disadvantages of using BAC vector system. One of the disadvantages is the construct that we obtain. It has large genomic fragment. Large genomic fragment may have non-related genes also that can lead to indirect or non-specific gene expression and unanticipated changes in the cell phenotype. Another disadvantage is recombinant BAC construct can be time consuming, and the large size BAC DNA construct that we obtain are easily degraded and sheared during manipulation before transfection. Then various applications of using BAC library. These BACs are being greatly used in modeling genetic diseases, that is to identify genetic disorders in order to study their effect in the experimentation on transgenic mice. BAC have been used to study neurological disease such as Alzheimer's disease or in the case of Down syndrome. The genome of several large DNA viruses and RNA viruses have been cloned as BACs. These constructs are referred to as infectious clones. BAC are also help to determine complete genome sequence of a given organism and to construct physical.
Thank you guys. This is all about back library construction. In the next video, I'm going to cover the another library, that is YAC library, yeast artificial chromosome library. Please do subscribe, hit the like button and do share the video. Hope so you understand. Thank you.